Okay, so in the first live lab meeting on Zoom, we will be mostly talking about the class and the syllabus. I won't plan on spending time on what I'm covering in this video, except maybe to answer questions from people to manage to actually complete it by then. And yes, I will be happy to answer your questions. I'm also anticipating that some class members might not get to this video by then, so I'm making allowances just for this first meeting of the semester. This video is on the biology-focused content of our first lab. We usually have 3 hours for a normal lab session, which is plenty of time to do the syllabus and some biology as well. This is the biology part. In the next half hour or so, you'll learn about our class experiment. And there's also an accompanying lab worksheet that I've posted on Canvas for you to complete and submit by the start of lab next week. In week 6, we'll be holding a more detailed discussion. That's a week when I'll actually release the data, and at that point we'll talk about the write-up for this experiment. One of the pluses of this weird remote teaching thing is that because we're not able to actually run a real experiment with live plants, this actually frees us from the constraint of having to wait for the plants to grow, usually 9 or 10 weeks, before we can even start to think about the write-up. Now, I have no problem being patient for good results, but in the layout of material we need to cover in lab, we have more discretional time earlier in the semester. So being able to get the experimental results in week 6 rather than week 10 will be just great. And you'll also get to be done with your papers earlier rather than having them add to that big pile of work you have late in the semester. This is definitely one aspect of remote BIO202 that I'll be missing once we go back to real experimentation, whenever that happens. What I'll do in this video is cover the basic biology of mycorrhizae in a bare bones sort of way, because you'll be doing some research on your own as part of this assignment, and I don't want to do all of your work for you. So the term mycorrhizae contains myco, which means fungus. A mycologist is someone who studies fungi. And it also contains rhizae, which means roots, as in the roots of a plant. Mycorrhizal associations are those between a plant and a fungus, in which the fungus connects up with the roots and there is some exchange of materials between the two participants. Soil is jam-packed with all different kinds of microbes, mostly bacteria and fungi, which are super important as decomposers. Now, plants put the roots through this hot mess of microbial stew pot, and they mostly have to fend off being invaded by the soil microbes. After all, a live plant is made up of all the stuff that decomposers normally eat, just livelier. Now, most of the soil microbes are either decomposers and or potential plant pathogens, but a few of them are considered beneficial because they actually help the plant with something. In the case of mycorrhizal fungi, these are the kinds of fungi that form connections with the roots of plants. Having them attaching to your roots provides an opportunity for material to be exchanged between the plant and the fungus. The plant can be a source of food for the fungus, mostly carbohydrates, sugars, which a plant produces through photosynthesis in its canopy above the ground. Remember this chemical reaction? And the sugar then gets transported to other parts of the plant, like the roots, where there is no photosynthesis going on. The fungus can tap into that sugar resource through the connection that it forms with the plant's roots. If it ended here, you'd have to call the mycorrhizal fungus a parasite, because it harms its plant by taking away sugars that should have gone to serving the plant's root system's needs. If you had one plant growing with the fungal root parasite and one growing without, the one without would do better because it's not feeding the fungus. Parasitism is when the host species, here the plant, is negatively affected by the association. But when we talk about mycorrhizae, we're usually thinking about a beneficial fungal partner. In other words, a mutualist rather than a parasite. In order for the fungus to qualify as a mutualist, it must help its host, In other words, if you have one plant growing with a fungus and one without, the one with the fungus would do better because of the benefits provided by the fungal partner. Mutualism is when the host species is positively affected. In order for this to be the case, the fungus must give back enough benefits to the plant to outweigh the negative effects of whatever carbohydrates or other things that it is taking away from the plant. Now, in the standard narrative, the plant takes benefit from having its roots infected by the soil fungus because of an increased capacity to take up water and mineral nutrients from the soil. You probably already know that nearly all of the water consumed by a plant comes from the soil through its root system. Additionally, the intake of mineral nutrients like nitrogen, potassium, phosphorus, magnesium, calcium, and a whole bunch of others occurs through the various salts that are dissolved in the water that the plant takes up through the roots. Yes, I'm basically talking about plant food. Now, in nature, plants get these nutrients from what you have left over after the microbes decompose all that organic matter from all the dead stuff, decaying plant and animal and microbial matter, and poop, of course, that are part of the makeup soil. These minerals are essential nutrients for the plant's growth, and they must be taken up through the plant's root system. The uptake of water and mineral nutrients is limited by the total surface area of the plant's root system. How much absorptive root surface is actually in contact with a moisture and mineral salt-laden soil. And although a plant's root system may seem like it presents quite a large surface area, it is really pretty paltry compared with the surface area of a soil fungus, whose filaments spread through the soil far more completely and are much thinner than even the finest root hairs that a plant can produce. The basic idea here is that the fungus can benefit the plant by producing a much greater absorptive surface area, thus facilitating the uptake of water and mineral nutrients. It's actually a bit more complex than this, but remember, I'm only doing bare bones here. I will clarify by adding, though, that the kind of mycorrhizal connection in which the fungus penetrates the root of the plant is just one of several distinct classes of mycorrhizal associations. This specific kind goes under several names. One is arbuscular mycorrhizae, or AMs. This name comes from the microscopic structures that form inside the cells after they've been invaded by the fungus. They look like tiny branching trees, hence the name arbuscle, which means little tree. When you put "ussel" at the end of something, this is a Latin way of making a diminutive. Arbor is tree, so arbuscle is little tree. Corpuscle is little corpus, or little body. And even the word muscle means little mouse. I'll leave this last one for you to figure out. Anyways, these same fungi also form round vesicle structures inside the roots, in addition to the arbuscles, and so they're also known as vesicular arbuscular mycorrhizae, or VAMs for short. The third name you'll find used for this same group of fungi is the endomycorrhizae. This name is also descriptive because it points to the fact that the fungi actively penetrate the inside space of the plant's roots. This is the endo part, as opposed to what occurs with another kind of mycorrhizae, the ectomycorrhizae, in which the fungus forms a filamentous sheath around the root without much penetration of the root itself. This particular distinction between endo and ecto is also significant in separating two groups of fungal organisms that are only very distantly related. The endomycorrhizae, which again is synonymous with arbuscular mycorrhizae as well as vesicular arbuscular mycorrhizae, are in their own phylum, the Glomeromycota, within the kingdom fungi, and are not closely related to the ectomycorrhizal fungi, which are mostly classified together with more familiar kinds of fungi, like the ones giving us things like mushrooms. In fact, many of the mushrooms used in gourmet cooking are special because of how they're found only in association with particular kinds of trees. Porcini mushrooms are only found near hazelnut trees. Matsutake mushrooms are found only near pine trees. In Japanese, matsu means pine and take mushroom, so matsutake literally means pine mushroom. These mushrooms are the fruiting structures of soil fungi that can only grow and thrive when they have the appropriate kind of plant host to provide them with the sugars that they need. Now, in contrast to the ectomycorrhizae, where there's often a very tight association between a particular kind of fungus and a particular host species, endomycorrhizae are much more generalist, much less tied to a particular species of host. They can and do invade the roots of many kinds of plants, thus making it literally true that the fig tree, pear tree, and banana plant growing outside my window are interconnected through an invisible fungal mycelium permeating the soil. If any of you are fans of Star Trek Discovery, this is quite obviously the working inspiration for the concept of a mycelium connecting all parts of the universe, which allows the Discovery to instantaneously translocate anywhere because its resident astromycologist has figured out how to navigate the ship through the mycelium. Probably too nerdy for most of you, but I thought I'd put this out there for that one person in a class of 30 who actually follows the show. Back to the main story. We now know that the majority of flowering plants can be suitable hosts for a relatively small number of known species of endomycorrhizae. We also know that these same endomycorrhizae have the ability to assist the plants with the uptake of water and mineral nutrients. You put two and two together with the mindset of how can I make money from this? And you'll realize that if you could formulate a product that provides a sure supply of the right kinds of endomycorrhizae for plants, this is something that gardeners would buy. And bingo! This is why we're able to manipulate mycorrhizae in a lab experiment relatively easily. All it takes is a quick search down the aisles of your favorite online gardening retailer. Home Depot or Lowe's would work, and you'll find plenty of products that contain the spores or live propagules of endomycorrhizae. And so here's where we come to our experimental design. Think of it as two experiments, M and N, going off simultaneously. In the M experiment, M is for mycorrhizae, we'll be manipulating presence-absence of live mycorrhizae by using a plant growth medium, aka potting soil, that comes pre-inoculated with mycorrhizae as well as other beneficial microbes. It's always a proprietary mixture, but they will list the main ones on the packaging, and it always centers on a few species of glomus, which is a glomeromycete. We always use the same product, this product, for both the M+, live mycorrhizae present, and the M-, live mycorrhizae absent. Difference being that for the M-, this growth medium has been put through a steam sterilization in our autoclave before the seeds are planted. This treatment kills off all the spores, so you wouldn't have any of the live microbes that are presumably there in the M+ treatment, which is not autoclaved. Here's a good question for you. Why is it better to use the same medium for the M+ and the M- treatment, but with steam sterilization on the latter, rather than having the M- plants growing in a different sterilized product that doesn't have the added mycorrhizae? Answer this in the associated lab worksheet. As far as our experimental predictions go, this one should be pretty straightforward, yeah? Figure that we start off all the pots with seeds of the same size, and at the end of X number of weeks, we'll weigh the plants after removing them from the growth medium. And presuming that we work from the basic hypothesis that mycorrhizae are good for the plants that they infect, what is the basic prediction that you would make in regard to the average plant weight for the M+ and the M- plants after X weeks of growth under otherwise identical conditions? Identical, that is, to the extent that we can achieve in our greenhouse. Write your prediction into your lab notes and also onto your worksheet as your answer to question number 2. In the N part of our experiment, we'll be considering the effect of nutrient supplementation. That's what the N stands for. Creative, huh? Basically, we'll have an N plus treatment in which plants receive a normal dose of fertilizer, while in the N minus treatment, plants will receive only a fraction of the recommended dosage. That growth medium that we're using, it's not like a normal potting mix that comes with fertilizers already added. Ours is marketed for professionals who apparently prefer there to be no fertilizers there at the start so that they can control all of their plants' nutrition completely. In any case, if we didn't fertilize at all, the plants would just sprout and barely grow and die when they ran out of nutrients. In our experiment, we use the same fertilizer, this product, at the recommended strength for N+, but at around 1/8 of that for the N- treatment. The second set of predictions, specifically for the N experiment, is also very straightforward. Presuming that fertilizers help plants grow, the plants in the N+ treatment should grow to a larger size than plants in the N- treatment, right? So far, so good, I hope. If we ran these two experiments separately, there wouldn't be much for you to think about this week. But we're not running them separately. We're applying a 2x2 factorial design structure in this experiment. 1/4 of the plants will be in an M- N- treatment, 1/4 in an M plus N-, 1/4 in M- N+, and 1/4 in M plus N+. If we have a total of 120 plants, this means 30 plants are there in each of the 4 treatments. This is why we usually need to dedicate a full lab period to data collection, something we don't need to do this semester because we're being forced to recycle results from an earlier semester. But what does this design give us that's different from having 2 separate experiments? Good question. In our minds, we're looking at both mycorrhizae and nutrients as things that should increase plant growth, right? But is the effect of each independent of the other, or is there some kind of interaction that occurs between the two? You can think of this as being similar to the issue of mixing different kinds of pharmaceuticals. We know that some combinations of drugs are bad, while others are good. An example of a bad combination would be where drug X more or less cancels out the effect of drug Y, like the way that the antibiotic tetracycline cancels out the effect of penicillin. You don't ever administer these 2 antibiotics in combination. Basically, penicillin works by weakening the bacterial cells when they undergo cell division, while tetracycline stops the cells from dividing. It's not like this combination does something that's bad, although there are plenty of examples of drug interactions that do result in some toxicity. But it's still easy to see why this is a bad combination. Tetracycline stops cell division, so there's no effect of penicillin. You don't get any more effect from the combination of both drugs administered together than you do from tetracycline acting alone. An example of a good combination of drugs would be any of the HIV treatments in widespread use today. All of them combine different antivirals that would have very limited effect if used singly, but in combination with the other components, they create an effective combinatorial treatment for HIV, something that eluded medicine throughout those dark years of the AIDS epidemic in the late 1900s. Okay, so for our plants, do you expect mycorrhizae and nutrients to interact positively, in other words, a synergism, or negatively, an antagonism? Please don't guess. This is not a situation where you can flip a coin to formulate your hypothesis. You should follow a reasoned approach in which you can justify your hypothesis on some basis that you can defend. You have the internet at your disposal for doing some research. It shouldn't take much, maybe a half hour or a little bit more, to look up what kinds of studies have been done and what results they got. This could definitely inform your thinking on whether you expect there to be an interaction at all, and if there is, in which direction, antagonism or synergism, it might go. Another way some of you might approach this is by integrating your personal experience, what you know about plants and soil fungi, even the things you learn in today's lessons, to work out a reason why there should be no interaction, or there should be an antagonistic interaction, or there should be a synergistic interaction. Now, even if you take this route of reasoning things out with just your own brain, it would still be wise to incorporate the findings of past studies into the forming of your hypothesis. I'm making this the third and most challenging part of the worksheet assignment. What do you hypothesize in regard to the interaction between the two manipulated variables in this experiment, mycorrhizae and nutrient supplementation? And by what reasoning do you come to this? You'll want to include your sources with your answer. And what were the findings from these past studies that led to your hypothesizing the way that you do? On Canvas, I provided a link to the worksheet for this lab activity as a Google Doc. The questions are the same as what I've given verbally in this video. Now make a copy of this worksheet onto your hard drive or into your own Google Drive, and then edit your copy by adding your responses to the 3 questions. Then format your worksheet in one of the acceptable formats, doc, PDF, there are a few others, and then submit it as a file upload to Canvas. You should get used to this method for submitting work for our class. We will use it a lot this semester. What you turn in should be your own work, of course. Plan on everything that you submit for BIO202 being evaluated for originality by Turnitin. This is something that compares the sequence of characters in your submission with everything on the internet, as well as everything submitted by others in the class, and for that matter, every student that has been in a class that has ever used Turnitin. And it flags those passages that are suspicious. Be advised that passages that are copied and pasted from someone else's work is very likely to be flagged even if you substitute words. Now this doesn't happen that much in my classes, but there's almost always a student or two who never learned that copying and pasting and altering is completely unacceptable as a writing practice. Okay, back to our experiment. Here's another way to think about what we mean by interaction. Let's say that the average plant size in the M-N- experiment, that's the one with no mycorrhizae and low fertilizer, is D grams, D for default. Now if little m is the effect, also measured in grams, that mycorrhizae have on plant growth in our experiment, then the M plus N minus treatments average is going to be D plus little m, the default size plus the added boost provided by mycorrhizae. In fact, we can use our experimental results to estimate the value of little m. The average of the M plus N minus plants minus the average for M minus N minus will be our estimate for little m. Now, if little n is the effect that nutrient supplementation has on plant growth in our experiment, then the average weight of the M minus N plus treatment should be D plus little n. And we can use the average of M minus N plus minus the average of M minus N minus to estimate the value of little n. Okay, in a completely additive world where mycorrhizae and nutrients act independently, you would expect the effects to add up. So if we look at the average for M plus N plus, this should be D plus little m plus little n. Now what if the two factors had a synergistic interaction? Well, with a positive interaction, the average plant weight for M plus N plus will be more than what you would expect under additive effect. And if there were an antagonistic interaction, the M plus N plus treatment would be smaller than the expectation for additive effect. I'll let that sink in a little, and we'll come back to discuss this further in week 6 or before. Remember that you could raise questions at any time.